So hi, everyone. Thank you so much for inviting me. I'm really, really excited to be here, although I'm really disappointed I couldn't join you in person. And I'm only joining remotely on a screen. My name is Sangeeta Bhatia. I'm a postdoctoral researcher at Imperial College London. I have been doing outbreak analysis for the last 5 years now. And I'm excited to share with you our work on using digital disease surveillance data for outbreak analysis. So that's what I'll talk about now. So before I start, let me just say that everything that I talk about now has been published in this paper. And it was a massive team effort, as is everything else in science. So all credit goes to my team, and anything that's wrong reflects on me. So with that out of the way, let's talk about digital disease surveillance. So when we talk about surveillance data are absolutely crucial to answer any kind of public health question that we have. So when we have a new disease, a new pathogen, emerging pathogen like SARS-CoV-2, or the re-emergence of a pathogen like, for example, monkeypox, public health decision makers need to know, like, is this a new threat? And if it is, like, how bad is it? And they need to understand its public health impact. So that ties into the question of severity and risk profile. Who does it impact most? What are the at-risk groups? Who do we need to protect? What do we need to watch out for? And they need to understand things like, how does it spread? What is the transmission mode? So they can plan appropriate response. Then as an outbreak spreads and enters into an acute phase, then we have forecast of key indicators like cases or deaths or hospitalizations are used to raise situational awareness, but also to plan public health response. And at the same time, we need to monitor the changes in transmissibility to assess whether the interventions that we have deployed are actually working. And none of these questions can be answered without surveillance data. If we don't have surveillance data, by which I mean not just case data, not just on the number of cases, but also other aspects of disease spread, for example, changes in population behavior or appetite for and compliance to public health measures, or things like vaccine uptake. So if we don't have data on these, we are absolutely flying blind and we can't answer these questions. So we need surveillance data, and digital disease surveillance has emerged as a useful complement to traditional surveillance, because traditional surveillance is really, as you can imagine, it's resource intensive, right? It requires a lot of investment in personnel, in money, infrastructure. And while we cannot, it's the pillar of public health, but digital disease surveillance can be used to complement traditional surveillance. And my goal here is to illustrate to you one case study where we have seen this. Right. So when we talk about digital disease surveillance, it can actually mean a number of different things. So for example, we have data that are collected digitally that relate to our digital lives. So things like, for example, the reports on population mobility that were released by Google, that have been released and continue to be released by Google and Apple during the pandemic. So these are things that are in the public domain. But there are also things like mobile phone usage data, which typically had to be purchased. They are commercial data, but both these kinds of data have been used to understand disease spread. So that's one source. Another source that you can think of here is, for example, monitoring changes in what people are talking about on social media like Twitter, or what they are searching for on Google, as to pick up a signal for disease activity. You can also have data that are collected through active or passive surveillance, but they are shared with us digitally via, let's say, a dashboard or as a PDF file. So in some instances, for example, with the COVID-19 dashboard developed here in the UK, you would have the data underpinning those dashboard and these fancy graphics. You can download them and analyze them further. But in some other cases, you only have access to the outputs of these analysis. So the analysis has happened elsewhere and you can only see the graphics and the results. A very useful source is unstructured data. So for example, news media articles. So especially during this pandemic, a lot of effort was spent by research groups across the world in the early phase of the pandemic to collate data that was being reported only via news media at the time. So I remember in the early days of 2020, here at Imperial, our team was spending hours every day tracking down every single case that was being reported in news in different parts of the world. And all of those data actually turned out to be really useful because it fed into critical analysis that we had done early, like assessing transmissibility and then initial estimates of IFR. So this is really useful. And then almost by definition, if you have a new outbreak, before surveillance systems can really kick in, news media would almost be, by definition, be the first to get there and report on that. So we will, in any public health crisis, we will have this unstructured data that we really need to tap into. And it would be extremely useful to develop automated ways of extracting these data. But what I'm here to talk about is this kind of multimodal collection. So ProMED is a prime example, and this is what I'm going to talk about here. In case you don't know how ProMED works, ProMED have a network of clinicians all over the world who would email them any reports of any unusual health events on plants, animals, or humans. And these reports are then curated and verified and vetted by subject matter experts at ProMED. And only then are they posted onto a network of ProMED subscribers. So ProMED has this really unique bottom-up surveillance model, which has worked really well. It is really a very successful example. So I'll show you in the next slide. It's really not important to read all this text, but the thing to bear in mind is that ProMED have really acted very well as an early surveillance model, and they have been able to raise alarms about various public health threats like Zika, and most notably, they were one of the first groups to raise an alarm about the cluster of pneumonia cases in Wuhan, which later became, novel coronavirus. So, right. So if this is an example of a ProMED email, and my objective is not for you to read this text, but the idea here that I wanted to show you is that ProMED, you get this email and it has a lot of useful information in it. There's a lot of quantitative information in here. So the scientific question that we had started out with was, how do we use this information? How do we go from this text, this textual piece, which has quantitative information embedded in it, to something which has like actionable insights? For example, if there is an infectious disease spread in a country, how can we understand how can we quantify the risk of disease spread? And how can, for a given country, how can we understand or characterize or quantify where is that risk coming from? So we wanted to go from something like this, from a ProMED email, to something like this. Can you see my mouse?
Yes.
Okay, cool. Yeah, so we wanted to go from something like this to something like that. So in 2018, our group here at Imperial, we got together with ProMED, with HealthMap, with Health Sites, and other partners to build a framework for doing this. So what we wanted to do was to get the number of cases over time and space from ProMED or HealthMap, combine that with information about a pathogen, right? So anything that you know about the pathogen, like its transmissibility, combine that with other sources of information, like how people are moving in a region, and things like, the health system capacity of a country that we could get from health sites, and put all of that together into a probabilistic framework to characterize the relative level of risk across space, and for each country, where is that risk coming from? That was the goal of our project, which we had called EMRIDS. So what we did when we got together, when we got that grant, as a proof of concept, we decided to do this analysis for the West African Ebola epidemic as a case study. And this is like, we got data from ProMED and we did all of this analysis. But just to get started here, what you're seeing on the left is data from ProMED that we got. And what you're seeing is a pre-processing workflow. So we had like a time series of suspected and confirmed cases, and there were some gaps, etc. And then we did some pre-processing, and we came up with a incident series, a relatively complete incident series. So the challenges that we faced were that data that we got were often noisy and required significant pre-processing. They could be biased. I mean, we need to be aware of that, and we'll talk about that later. But the more important issue here was that we could have like missing or incomplete or duplicate data. And there was some manual intervention that was required from collecting this data to analyzing this data and generating these insights. So I will now illustrate this pre-processing workflow to you. But before I do that, let me just emphasize that the challenges that we faced with this data set are not at all representative, right? Your challenges, if you are using digital disease surveillance data, may be completely different. So the takeaway of this exercise is not really a specific technique or a specific method, but my goal is just to highlight the kind of challenges that you can face in practice. And then I also want to highlight that there's some kind of inherent tension between building reproducible and very generic analysis pipelines that you want to do as an analyst. And then we have these data that will, every time you get these data, they will throw some different challenges at you. And not only will the challenges vary from one data source to another, they will also vary from one data cut to another, even if you're getting data from the same source. So there's some kind of tension over there. And I'll show you in a minute and you will see what I mean. So just bear in mind that the challenges are not meant to be representative at all. Okay, so let's do that. Before we get there, let me show you the data. Let's get rid of this. Right. So in the paper that I show, can you see this comfortably, or do you want me to make it bigger?
Bigger, please.
Okay, I'm going to try. I don't know how to zoom in. Okay.
Just bigger font.
Is that okay?
Yeah, much better.
Okay. All right. So in the paper that I showed in the beginning of my talk, we had used 3 data sets. So there was a ProMED data set, that the data set on West African Ebola epidemic that was curated by ProMED. We had another data set that was curated by HealthMap. And then we had a data set that was shared by the WHO. So WHO had organized the international response to that epidemic. And they had this data, which they shared with the research groups who were supporting the response at the time. But the data themselves were not in the public domain at that time. They were made public later. So what the data set that we have from WHO is really a significantly cleaned and pre-processed data set. So the reason we are using is like we will be benchmarking our ProMED data against the WHO data. That's the idea. So let me just show you very quickly the raw data before we have done any processing. So this is ProMED data. Sorry, I should also mention that this is what we did in the paper, but for the purposes of today's exercise, I will just be using ProMED and WHO data. Challenges are like more or less the same. And I also have a RMT file that's in the repo that I have shared on Mattermost. So if you want to follow along, you can check out the RMT from there. Okay, so this is what ProMED data looks like. We have stuff like location, country, region. So clearly you can see that there was an idea here to collect this data at a subnational level, but from the overlap in the data in this column and in this column, and the very many NAs in this column, you can see that we had a lot of missing subnational data. So we could only analyze this data at a country level, not at a subnational level. All right. And then, as I said, ProMED reports on many diseases. So this data was curated for us. So it only relates to Ebola and in humans. Then we have some metadata, right? Some like, where is that coming from? What's the alert? Stuff like that. Then this is the thing that we are interested in, right? So this is new suspected cases, new suspected deaths, new confirmed cases. So this is a daily time series. And you can see it's like really incomplete. And then these columns are suspected cases, suspected deaths, confirmed cases, and confirmed deaths. And these are the ones that we will be really interested in. Sorry, give me 1 second. Okay, so this is what this data looks like. Then we have WHO data. So the WHO data was really a line list. So a line list is each row in the data set is a case. So if you want to know how many cases were on a given day, you just count the number of rows for that day. And we'll do that in a minute. So they're slightly different. For the WHO data, you can see that it's a lot more complete, obviously. And even we have administrative 2 unit coded in, but because we want to benchmark ProMED data against this data, we will not be using this, and we will also analyze this data at a country level. Okay, so this is what the data looks like. Now we will go and do the analysis, right? I will make this bigger for you. So the code is on the left and the console is on the right, which is, if you are used to RStudio, what you would see in the bottom panel here is on the right. Okay, so I'm just going to load all the libraries that I need. I just like to do it at the top. And then there are some functions that we wrote to process these data. I will talk about them when we actually use them, but let me just source them so that they are available. Okay. So the West African Ebola epidemic, majority of the cases came from 3 different countries: Sierra Leone, Liberia, and Guinea. So we'll focus our analysis on these 3 countries. So let's, this is just useless. Okay, let's read in the data and just filter on the fields that we are interested in, the specific disease in humans for these countries. So if I look at what that looks like now, this is exactly what I showed you there. We have location, country, etc., etc. Yeah, I'm just going to use a janitor package to make this easy to analyze. It will just make the column names snake case. So we have not done any processing yet. All we have done is cleaned in the column names. This is just because the code that I have written uses the specific column name, so it's really not important. Okay. So what I want to do first of all here is I have this issue date, and you can see very clearly that it has time and it seems to be automatically generated. I'm not really interested in the time. I only care about the date. So what I will do is I will get rid of the time from this date column. I don't care about it. So I've done that. Okay. So now we are actually ready to look at the data, right? So far, we have not done any processing. All we have done is fix these dates. That's all we have done. So we are going to look at remember the suspected cases and the confirmed cases column that I have shown you. Those are cumulative cases. So that's what we'll be looking at. All right, so let's look at it. This is just me writing some code to using ggplot to analyze, look at these. So let's look at what it looks like, right? Let me bring it here. Right. So this is what the raw cases look like for Guinea, for one country. And right off the bat, you can see that there are problems with this data, right? There's a lot of missing data over here. We don't really know what's going on. And this is a cumulative time series. So it's really supposed to be increasing, but clearly it's not increasing. There are, like, you can see these issues here, what's going on here, this is going down, that doesn't make any sense. And that's just for one country. If I look at the other country, right, similar issues in this around the same time, in the same part of the time series, we have these gaps that we don't know what to make of. And then there are these inconsistencies that we're not sure about that seem to be errors. If you look at third country, we have similar issues. So right about the same region, we have blanks, and then we have this like, what's going on here? What does that mean? Right. So we can see that this data is going to need some kind of cleaning. That's not the most exciting part of analysis, but it's really vital and it's the foundation of any scientific project. Okay. So what I'm going to do is I will split this data by country, and for each data set, I will apply the same kind of pre-processing workflow. So that's how I like to work. So we had this raw data set. I'm going to split it by country so that I get a list of 3 data frames by country. And the first thing I'm going to do is we had the suspected cases and the confirmed cases. So just to keep my life simple, I'm going to get the sum of suspected and confirmed cases as the total cases. Okay, so you could see that there were a lot of NAs. If I scroll up, you will see that if I just look at the suspected and confirmed cases, there are a lot of NAs. So I want to add them up sensibly. If they are both NAs, I really don't know what's going on. But if one of them is NA, I want to add after removing NAs. So I'm going to do that now. This is what I'm doing. If I now look at what's going on, just as an example, right. So I have created a cases column, and again, there are NAs, right? If you look at this example here, you can see, so what we were expecting was that for each date, we will have a row in our data set that will tell us how many cases there were. But you can see that for each date, for a single date, we have 3 different rows, and the 3 different rows are giving us 3 different numbers. If they gave us the same number, that would be a really, really easy case, because if we have 3 different rows, it doesn't matter. They are telling us the same thing. So I'm just going to keep one of them, and I'm keeping consistent information. But here, for the 22nd of March in 2014, one row is telling me that there were 6 cases, and another is telling me that there were 80 cases. So what I'm going to do is I'm going to merge these kind of duplicate rows in a sensible way. I just have to make up rules to process this. So what I want to do is, for a given date, if I have duplicate numbers, I want some kind of principled way of combining them into a single row. And the way that we have selected here is just, if you have multiple data points for a given date, pick the median of those and keep that. Yeah, I don't know, it's just, let's do that. It may not make a lot of sense. It just requires for you to stare really, really hard at your data set for very, very long and see what makes sense in your case. This may not be the most sensible thing. And with more time, maybe we could have done something better and more informed, but that's what we go with now. Okay, so that's what we have now. So if I look at the same data set again, I have, oh, sorry, I'm supposed to look at this. Right, so what I have done is I have made my data set in some way consistent so that for each date I have only one row. Okay, so then if you remember the graphs that I showed you earlier, there were some kind of outliers. So we want to remove them, but we want to remove them not because they're outliers, but because it seems like these are errors in the data, because we are working with the cumulative time series and that's only supposed to go up. It's not supposed to go down. It's not going to make any sense. So we see these data, but we want some kind of principled way of doing it so that we can do it in a reproducible way. And then when we get a new data cut, we don't have to go and manually edit our data. So there are outlier, like this kind of stuff is a whole big thing in itself, but we use something very simple. So all we did was we said, okay, let's look at the last 20 days and 20 days of data for each data point. Let's look at the last 20 days of data. And if my point lies certain number of standard deviations away from the mean, that means that it's probably an error. So I'm going to remove it. Again, it may not make sense for the data set that you have, and you might end up removing like useful or crucial information, but just working with our data set, we found, okay, that makes sense and we'll go with that. So we'll do that now. Let's get rid of that. So we've done that. Okay. So we have got rid of one problem. The other problem that we had was that this was not increasing, and we want to make sure that this is an increasing time series, right? So we are going to do that. So how do we do that? So all of these functions that I'm using are actually in the files that I had sourced in the beginning. Let me open one of them and show you. I think it was processing. Okay. So for example, here, so this is what we are going to use. So we want to make sure that our time series is increasing. So what we'll do is we'll look at where it is not increasing, and we will say, if I'm at time t, and suppose t and t plus 1 are not in increasing order, then if I remove t, does it become increasing? If so, I will remove t, the data point at time t. If I remove t plus 1, does it become increasing? And so then I will remove that. If removing both of them or none of them makes my data set increasing, my time series increasing, then I will remove both. Again, this is like really kind of intuitive or hand-wavy way of working with this data. Probably not the best, but that's what we did at the time. And it made sense when we looked at the results of the workflow. Okay, so that's what we'll do now. And that's what we have done. For each country, we have made sure that the cumulative time series is now increasing. And if you remember, there were like a lot of gaps in the middle. So we want to fill that data. We want to fill those in missing points. And again, there are any number of ways in which you can do it. It can go from something very simple, which is what we have done here, to something very, very sophisticated. You can have a model-based way of filling that data. But what we did here was something very simple. We said, okay, this data is supposed to be like, it will look like an exponential. So it will be a line on a log scale. So I'm just going to use that kind of log linear interpolation to fill that data, and which is what we did here. So I'm going to do that now. Okay, so we have done that. So what we are now supposed to have, because we have filled in all the dates for which we didn't have any data, we should really have like a daily time series for each country. Just checking that that makes sense. Yes, the difference in dates is 1 for all of them. Okay, so far we were working with cumulative time series, but for analysis, we want to work with the daily incidence time series. So we will go from the cumulative to the daily time series. And that's really simple. You just, to get the number of cases on a given day, you subtract the number of cases on day t plus 1 minus day t. That's what we'll do here now. Right, so I got these. Okay. So I have all of this. So what I'll do is I'm just going to illustrate to you the figure that I showed you on the slide. I'm going to illustrate that. This is just a silly function that I wrote to plot them in a sensible way. So I'm going to plot each step of my workflow and to see what difference, how it has made a difference to my data set. So let me just plot that, plot all of that. Okay, so now, okay, so remember what we did? We started with raw data. From raw data, we went to which had suspected and confirmed cases. From that, we went to the total number of cases as a sum of suspected and confirmed cases. Then we removed any duplicate data on a given date in a very simple way. We removed any outliers, any kind of data points that didn't make sense in the cumulative time series. We made sure that the cumulative time series is increasing. Then we interpolated the missing data. And from that, we got the daily time series. So we are going to now look at what that looks like for one country. I'm just going to make this bigger. It's going to give me an error. Let me try that again.
How many points in this one?
How many points? Sorry, what's the question?
Yeah, I'm saying it's very slow. I'm thinking it's because it's a lot of.
No, it's only slow because there are so many things happening on my computer, not because there's a lot of data, right? So this is what we had. This is how we got the total number of cases. There were like a lot of inconsistencies here that we got rid of. When we got rid of, we had this massive gap, which we have filled, I think, in a sensible way. It doesn't look, I mean, you can imagine that if we did actually have data, this is what it would look like. And then from that, we got the daily time series. Okay, so let's go back. So what we are now going to do is we will compare this data with the data that was collated by WHO. And remember, before we do any comparison, remember that the data set that I'm now using is really extensively clean data set. And if you were actually analyzing it in real time, which I'm sure many of you would know, this is not going to be that clean, and it will have like massive issues that you will spend hours dealing with. But we have this clean data set and we can work with that. So I'm going to read the same. I just keep the columns that I'm interested in. Okay, so as I said earlier, this is a line list data, and what we want to work with is an incidence time series. So you just want to, for each date, count the number of rows in your data set. That will tell you how many cases you had. There's a package called incidence, and there's a utility function that will convert a line list to a time series. And it just needs dates. So that's what we are going to do. So I'm going to do it again by country, and then convert the line list into a time series. Okay, this is just so that both my data sets have the same column name so that I can plot them together. Sorry. Okay, so now I have this data set, which I didn't have to do anything with. I just read it in, and I have an incidence time series, and I have the processed data set from ProMED, and I'm going to compare the two. So I put them together so that I can plot them, and I can color them by the data source, which is either WHO or ProMED. So I'm going to plot them now. Okay, so let's go and look at the data. Okay, so I hope you can all see this. So the red dots, the legend is on the top, so you can't see it, but the red dots are ProMED data and the blue dots are WHO data. And I mean, if you squint really hard, you can see that there are some areas of agreement. So especially in the early phase of the epidemic and in the tail of the epidemic, they broadly agree with each other. There is greater discrepancy here when you have a peak, but you can see that like part of the discrepancy is coming from the way that we have filled it, at least, right? This is like interpolated data, and maybe we used a very simple approach. We could have used a better approach. It is also possible that this particular data point is probably an error, and it might be worth checking what's going on here so that we are completing this curve. in this way. Yeah, but if you, yeah, but there are like, you know, agreements over here and over here. Similarly here for Liberia, you can see that these points are really distorting the scale. If we didn't have these points, then clearly the broad trends in the two data sets are in agreement with each other. And similarly for Sierra Leone, these points are like just distorting my scale. If I didn't have these, then the two time series would be, I can see that they are in agreement with each other. So there are areas of agreement, there are areas of disagreement. We'll see what we can do with that. Are there any questions at this point? No? Shall we just carry on? Okay. So we have these two time series and we stared really hard at it and we said, okay, fine. There are some places where they agree in the early phases, in the late phases, in the middle, like there's stuff going on, not quite sure what. Remember also, I didn't mention this before, but WHO data combine the suspected, confirmed, and probable cases. But for ProMED, we didn't have probable cases. So there's that thing to bear in mind, that there's like a source of discrepancy that we know why it's there. But other than that, there are some things. So our question, okay, so we are doing this retrospectively, and therefore we have this alternative data set we can use to compare ProMED data with or corroborate our data set with. So we have these two alternative data sources of information and we are comparing them. But if you were doing this in real time, you really wouldn't have that. You know that your data are like, you know, slightly messy, slightly incomplete, whatever, but you still want to draw some kind of insights from them so that you can understand the situation. Like, where is this outbreak going? Is it growing? Is it declining? What are we doing? What's going on? So to draw some kind of insights. So we will still use these data to estimate reproduction number. So reproduction number, the effective reproduction number, is defined is the average number of secondary cases that an individual infected at time t would generate if things remained as they were at time t. So if nothing changed, this is how many secondary cases, average secondary cases, an individual infected at time t would generate. And we will use a package called epidemic to estimate the reproduction number. Okay, so to estimate reproduction number, you need the serial interval. Serial interval distribution is the delay, is the distribution of delays between the onset of symptoms in a primary case and their secondary case. So it roughly tells you how long is the delay between these two events. So it tells you how fast something can spread. So I got these numbers from the literature for Ebola. So we have the mean serial interval, which is approximately 2 weeks, 14 days. We have the coefficient of variation, which is the standard deviation divided by the mean. And then typically, like in most cases, we will represent the serial interval as a gamma distributed random variable. But gamma distribution is really infinite. It has infinite support. It goes all the way to plus infinity, but that's not going to make sense in our case, right? So we know that that delay cannot really be bigger than a certain number. So we just have to decide, or from the literature, be informed by the literature, that the delay of between the onset of symptoms in a primary case and their secondary case cannot be bigger than a certain number. So in this case, we said it cannot be bigger than 40 days. And what we want to do is we want to go from this continuous distribution, which is a gamma distribution, to a discretized distribution over this time period, 0 to 40. That's what we want to do. So we will use a function from epidemic to do this. Let's do that and normalize it. Oh, sorry, what's to normalize it, to make sure that it adds up to 1 over this time period. So let's look at what that looks like. What did we say? 40, right? And then we want to see what is the distribution. Fine. So we look at, so this is what it is. So the x-axis is the day and the y-axis is the probability. So this is the delay in days and y-axis is the probability. So this is what our student interval looks like. Okay, so we are going to use this to, our objective here is to estimate the reproduction number for each country for each data source, and then compare them to see whether they are consistent with each other. If they are consistent with each other, they are telling us a consistent story. And that gives us confidence that we could use ProMED data for outbreak analysis. But if they are telling us like different trends, then that's really concerning and we want to see why that's happening. So what we will do is we had made this data frame, which put together data from WHO and from ProMED for each country. We have put it together. So I'm just going to split it again. I'm going to split it again so that I can do this analysis for each data set. So let's see what the what this is. Okay, so we have ProMED data for Guinea, WHO data for Guinea, ProMED for Liberia, and so on for all the 3 countries. Okay. Now, here is another parameter that you have to choose when you are estimating the reproduction number. So the time window. So this is the time window over which the reproduction number is estimated. And if you choose a very big time window, then you will get very smooth estimates, but maybe you will miss some temporal changes in that window. As against that, if you pick a very small window, then you will get really choppy estimates. So maybe you're picking up like, you know, spurious changes. So you have to maybe experiment with a few different time windows to get a feel for what's right for your data set. So let me do this for one data set first so that I can show you what the output looks like. So I have this all groups. Let me just get the first one. And I'm just going to execute this. Okay, so if I now look at the output of this, output of just what I have done, what EpiEstim will give me back is a data frame, and it will estimate, as I said, it estimates reproduction number of over a window, and that's a rolling window. So it goes from 2 to 16 and 3 to 17 and so on. And for each window, it's telling me the mean reproduction number, the standard deviation of the reproduction number estimated in that window, and the various quantiles. And this date I had to affix because that reproduction number is really kind of for that window. But I want to do a temporal comparison. So I have to say, I have to decide whether I'm going to assign the beginning of the time window to that estimate or the end of the time window or a date in the middle. And sometimes it will make a difference to how you perceive things or how you interpret them. So here I have affixed to this the end of the time window. Okay, so this is what's happening here. So I'm going to just do this for all the countries and all the groups and just process it a little bit. Okay, so now we are ready to visualize this. So I'm going to visualize for each country the reproduction number estimated using WHO data and using ProMED. Oh, sorry. Okay, so the blue is WHO and the red is data from ProMED. So again, as we saw in the incidence time series, we can see that there is broad agreement here in that we are getting consistent trends. So if reproduction number is below 1, that means that the outbreak is declining. And if it is above 1, that means it's growing. Yeah, we get kind of broadly consistent story here, right? Again, there is greater discrepancy in the middle, but in the tail and in the beginning, they are more consistent with each other. So what we have done, if I go back to my slides now, Okay, so what we have done is we have analyzed this data. We compared the data from WHO with data from ProMED. I'm just showing you a figure from the paper where we also did this for health map, as I said earlier. And we compared the time series and we said, okay, there are discrepancies, especially in the middle, but in the early phases and the last phases, they agree with each other. And more importantly, they tell us a consistent story about they give us consistent insights that we would have obtained from WHO data if we had access to them about the trend in the growth of the epidemic, which is a really useful thing that we need to know. We did some other things that I'm not talking about here. So for example, we used these reproduction number estimates to generate forecasts, and they captured the epidemic trajectory well. We did this, which was really interesting. So what we have done here is basically, as I said earlier, what we want to know for each country, where is that risk coming from, the risk of importation. So the relevant country is shown in gray, and the various possible sources of importation are other countries where Ebola outbreak is happening, or we have identified Ebola cases. And the scale of the risk assigned by the model is this. So if it's red, is high. And the black dot here tells you where the actual importation happened from. That was identified, using later epidemiological investigations or genomic investigations. So what we want to see here is basically, we want to see this black dot in an area which is red, because that's where the model assigned the highest risk. And you can see that, like for most of the countries, that's actually what's going on. The importation is happening from a country to which the model had assigned high risk, which tells us something, which gives us confidence that we can use these data. All of this is happening using ProMED data, by the way. So we can use these data to set up some kind of early warning system, surveillance system to do that. So that was an important insight. Okay, so to conclude, what's my takeaway here? We use ProMED data. There were some discrepancies, but we should not really be discouraged by that. And bear in mind that these kind of digital disease surveillance systems, especially things like ProMED that have this kind of manual curation at the end, are really, they will struggle to cope when we have a lot of cases. So in the peak of the epidemic, when active formal surveillance systems have been set up and they have kicked in, they might be less useful. But in the early phases of the epidemic, in an epidemic, and in the last phases of the epidemic, when you don't have formal surveillance systems, these kind of digital disease surveillance systems are incredibly useful and they have immense value. We know that data processing will present challenges, and these challenges will be really variable, differing from data set to data set. So it's really important for us to develop an ecosystem of tools into which we can have this plug and play kind of analysis pipelines. And automation of data collection to analysis pipeline will go a long way towards utilizing these, extracting the maximum utility from these data sets. So the value of these data, they are available, they suffer less from reporting delays. There may be other problems, but they suffer less from reporting delays. And that's something to bear in mind. So for example, for this pandemic, for this particular epidemic, I remember that the WHO SITREP, I think, came out in, the first SITREP came out in August, as far as I can remember, and that reported on the cases from January to August. But the report from ProMED had actually been published in January or March, but whatever, it was earlier than August, right? So this is something to bear in mind, and this is why it's important to use these data sets and to spend efforts trying to extract maximum utility from these data sets. And there's another important aspect that when we analyze this data and we understand the gaps in this data, we can also use the insights to inform data collection and therefore improve data collection efforts. So this is like a feedback loop. Yeah, those were all the things that I wanted to say about digital disease surveillance data. Happy to take any questions.
Could I ask you, so did you eventually understand what explained that discrepancy in, what was it, August 2015, no, 2014, between WHO and ProMED data?
Yeah, so as I was saying, there's like the probable cases thing. So that's one source of discrepancy because WHO data had probable cases of Ebola in them. They were included in the case count, whereas for ProMED, that's not the case. And there was also, we have several hypotheses. We have not been able to test them rigorously. Like, for example, there was this, ProMED data will be a little bit subject to, especially when the outbreak begins to spread, and then we start using like news media articles, etc. So there's going to be variation in reporting, and that will affect your numbers. So that's something that digital disease surveillance is going to be impacted by.
There's one more question from Ilona about the discrepancy between ProMED and WHO. Could be because there is a media hype.
Yes, exactly.
So
yeah.
How do you correct for media hypes? And
that's very true. Like, that's what I said. Like, there can be changes in reporting over time, and if you, basically there are ways of correcting for it, right? So you can have a. We didn't do it in this particular paper, but you can have like a variable rate of reporting, and you can either estimate it or you can kind of put some kind of prior on it based on your empirical data or understanding that at the peak of the epidemic, there is going to be a greater probability of reporting. So you can incorporate that into your model. It's just that it makes the model a lot more complex. And if you want to do this in real time, then you want to have a simple model that you can run, and get answers like in a day at least. So we use this data. We had the thing that I didn't show you is we had a spatial model, a spatially explicit branching process model, where we were jointly estimating the parameters of the transmission model, as well as of the reproduction number, as well as the probability of movement. And that just makes the model a lot more complex. And so we were doing this analysis week by week to mimic how we would have done it in real time. So in the first few, I think maybe months, the model would run really quickly. But then as we get more and more data and we fit the model to that, it just takes so much time that you would not be able to do this in real time. So the more you can have something like reporting probability, but it's just, you need to have some kind of way of informing that parameter.
okay, more questions? Elena?
You can talk. Hi, thank you for the presentation. It was very useful, very good.
Thank
so
you.
how much time did it take you to clean the data from the 3 data sets?
It took me forever. I mean, the problem was not so much writing the code or actually running the code. The time taking part was understanding the data and trying to say, okay, this is a problem and this is how we're going to fix it. So that was the time consuming. Understanding the data, interrogating your data is really more the most time consuming part. But once you have understood and once you have come up with like these rules, okay, I'm going to take the median of these dates, then it's really easy to write the code. That's really not the hard part.
Hi, thanks very much for that. It was very interesting. If you were to recommend to WHO or ProMED how to improve their data sets to make life easier for you, how would you do it?
Actually, we have been having these discussions with ProMED because we work really, like, for the last 5 years, we had been working closely with them. So I think the thing that we have focused on most and we would strongly recommend is having a like automated feed from ProMED. That would make life easier because once you have an automated feed, then you can write. Okay, let me explain what I mean by that. The data that I showed you, it was actually downloaded by ProMED colleagues, and it was shared with us via email. And we get like, maybe we got like 2 different cuts of the data set, and there were small discrepancies between the 2 data sets. So what would really help and what would really encourage the development of tools around this kind of ecosystem is having automated data feeds. Because once you have that, then you can develop general purpose tools that will always work with your data.
So you reckon the data is clean enough?
I don't reckon the data is clean enough. I'm saying that the data can be cleaned in a generic way. I don't have to write custom code every time I get data. That's the thing that's going to take time. I don't want that. I want to write I want to have a system where I can plug in my data and get some kind of output, and then I can look at my output and see if that makes sense. So what I'm saying is that even if the data are not clean, which is perfectly fine, I want to have automated ways of cleaning it, whether it's like changing column names or whether it's, dealing with like whether it's cases or something else. Like the thing that I showed you, like the suspected cases thing, it made a lot of sense in the case of Ebola, but it might not make a lot of sense in the case of a different disease like COVID. So even the column names that you would get from ProMED might be different. And those are the kind of things that really trip you up when you have written code. So for every data cut, you have to write custom code, which is really inefficient. So if you had automated feeds, then you can develop general purpose tools and you can clean the data to the extent possible in an automated way. You can look at the output of your analysis and then see what else is needed for this specific data set.
Thanks.
And was the country level data enough for you, or you think ideally you would like to have per admin level?
Definitely, we had a lot of conversations with ProMED about this, especially with COVID and other diseases. So we met in 2020 and we got some additional funds to extend this work. And one of the topmost things that our topmost priority was to collect data at a subnational level, because that really impacts, like, these countries are big countries, and how the disease is spreading within the country really has an impact on how it's spreading outside the country. So that was our topmost priority, but then, COVID happened and everyone pivoted to COVID. So we couldn't get to that.
This R markdown you send us, I mean, it's still just the RMD file. You didn't produce like MD file. Is there any special reason so that that it renders in the GitHub?
So there is a README on the top level, and that's a MD file. And then this RMD that I have produced is only so that you can just follow along with the analysis. So that was like, if you do it on GitHub, it's maybe not going to be very useful, I guess, because you want to be able to run it on your machine. But the overview, I have tried to give an overview and the links to the papers that I used in the README in the top level of the repo.
Okay, let's do it together. I'll share my screen so you can see.
Sure.
So here's my screen.
Yeah. So
tell
if you look,
me
yeah,
what-
this README, so this gives you the kind of, overview of what I was talking about.
Okay.
Yeah.
And then I see this, for example, in the analysis.
Yeah.
The RMD file.
Yeah.
And then also here in the data row.
Yeah.
That's just the raw data. So yeah, this thing I'm talking about, this one.
Yeah.
There's no MD file. You haven't made it.
No, I didn't make it. Like I didn't think that it will be important for people, I guess. I don't know. I mean, I just didn't think. Yeah.
I'm just checking if it's, maybe I'm missing something, but
No,
okay.
you're not missing anything. That's the only file I checked in.
Okay. And then this one is just these are just functions and
Yeah, the functions that I was talking about. So you can have a play and maybe change them. The things that I was telling you, right? The way we interpolated and the way we picked the median, maybe that was not the best thing to do. It could be done in a more sensible way, perhaps.
and there's this book from Paula Moraga. This book
On
let me see.
spatial. Is it on spatial?
Yes.
Yeah.
And she uses it a lot.
Yeah, we didn't use INLA. We set up this model in Stan. Maybe because of my lack of experience in INLA. Yeah, it's something
one.
that I understand. Yeah, it's like something I've been meaning to explore, but I've just haven't got around to that.
Okay. Well, I was just curious if you maybe use some of these like inline methods, because they seem to be quite generic and they're using the space-time framework. So you basically use the maps of, I don't know, this is the air quality somewhere here on the end.
Yeah.
So,
like I'm aware of these, but I just didn't get around to, yeah.
Okay.
All right.
Thank you so much.
Thank you so much for
Bye.
inviting me again. Thank you so much. Bye. Take care then.